I'm now going to introduce to you Dr. Said Danishmand, who's known in the industry as Dr. D. He's with San Diego Fertility Center. He's become a great friend. He's internationally recognised fertility specialist with a lot of experience in what we call third-party reproduction. He specialises particularly in complex IVF cases, and he's done research on endometrial receptivity as a way to maximise probability of success. He does a lot of talks around the world, and we're really lucky to have him today. His company's a platinum sponsor of the conference. He's got patients coming in from 35 countries to his clinics in various places in California and Nevada. Please welcome Dr. Danishmand to come and talk to us today.
Thank you, everybody. It's good to be here. It's actually good to be back in California. So let me see how I can... So this is what I want to do is kind of, this topic is a little bit heavy. So after it's done, I don't want you to kind of say, I guess that's what they meant by he drones. But this is Daniela. So the other day I was doing some emails about 1:30 in the morning and a little tired, kind of struggling to keep my eyes open. And then I received an email from an intended parent. And it was a story of Daniela, who was in the neonatal intensive care unit. It was a singleton gestation. The surrogate developed preeclampsia at 25 weeks of gestation, severe preeclampsia, and Daniela delivered. She was about 1 pound, 2 ounces and struggling for her life. So I received this email. Now she's 8 years of age. There's a great picture there of her first day at second grade, and she's on top of her math class, English class. So I just felt that this was an inspiring email. This is the reason why we do what we do, to create families and to hear these stories. So to me, that was great, and I kind of reignited my energy for that night, and I worked till about probably 3 a.m. So let's talk about something that is important. If I can get the slides to, there we go. So we're going to talk about transfer decisions, and there's 2 areas that I want to concentrate on, because obviously, again, as I said, this topic can be a little bit heavy, but the 2 areas are single versus double embryo transfer in terms of how that is going to translate into pregnancy rates, live birth rates, and success rates. And the other issue is something that you should all discuss with your physician when you have your initial consultation, and that is whether you should do chromosome analysis, PGS testing, PGD testing, because there's a lot of data out there and not consensus. So San Diego Fertility Center was established in 1989. They're international leaders in the field of surrogacy. About 60% of the practice is international, so we have patients from all across the globe. Carmel Valley, San Diego is the main center, the main clinic, and then there's clinics in Mission Valley, Temecula, and then I have a satellite office. We have a satellite office here in Los Angeles on La Cienega, and we also have one in New York City. Now, we use the New York City office for a lot of our European patients, intended parents who may not want to travel all the way to the West Coast. We use that facility as a place to do FDA screening and also to do sperm freezes, and then we transport the sperm over to our clinic on the West Coast. So in vitro fertilization, you know about the fact that what we do is we stimulate the ovaries with medications to mature and develop multiple eggs, and then the eggs are retrieved. This is just a photo, an image, a video of a embryo transfer. You can see the catheter going from screen right to left, and this is one of our transfers where the catheter goes in, the lining of the uterus is nice and thick, it's a trilaminar pattern, and we push the catheter a little bit farther up from where we want to transfer, and then we pull it back, and there it is. That little spot is where the embryo was transferred. That's not the actual embryo itself. The embryo is microscopic. That's the media around the embryo. And so that we can tell. Now, what we do in terms of determining whether a surrogate potentially can have a double embryo transfer or potentially can have or minimize the chance of twins like preterm labor and delivery is we actually measure the depth of the uterus when the surrogate comes in for her screening. And the depth of the uterus is an important measure because if the uterus doesn't measure very deeply, then there's a potential higher risk of preterm labor and delivery, which is really the main risk with a double embryo transfer. So embryos, as you know, are selected based typically on morphology, meaning that we let the embryos grow for 5 days, and then as they develop from 2 cells to 4, all the way to a stage called a blastocyst at 128 cells, what we do is then we look at them under the microscope, and we have certain objective criteria for embryos that we published on back in 2002, and then we select the best quality embryo for transfer. The embryos can be graded. What we've done is we do a simple grading system from grade 1 to 3, 1 being the best grade embryos. And this is, again, based on multiple different parameters. I'm going to show you a photo of a blastocyst so we can discuss what are some of the things that we look at when we select the best quality embryo. Now, some of the things that we can't determine morphologically under the microscope are chromosomes. So we can't tell if an embryo is chromosomally normal. But the interesting thing is that there was a study that was published, and what they did was they looked at certain criteria morphologically, how the embryo looked under the microscope, and they took the best quality embryos and they did chromosome screening on them. And what they found was that over 90% of embryos that looked really good under the microscope were also chromosomally normal. So we do believe that morphological analysis under the microscope is really important to pick the best quality embryos, and most of those embryos are going to be chromosomally normal. Most of them are going to develop into live births. But as you know, there's 46 total chromosomes in humans, and there's 2 copies of each chromosome, chromosome 1 all the way to chromosome 23. And many human embryos have chromosomal abnormalities. So when we look under the microscope, even when we use donor eggs, approximately 60% of the embryos that are created from donor eggs are going to be chromosomally abnormal. But the ones that are chromosomally abnormal typically don't make it to the blastocyst stage, and even if they do, morphologically under the microscope, they don't look normal. They have areas of fragmentation, the cells don't look normal, so we are able to make a good assessment as to the quality of the embryos by just looking at them under the microscope. So we can't, as I said, count the chromosomes in our microscopes. There's about 25,000 genes on all 46 human chromosomes combined. Some patients have a genetic abnormality in their family. For example, things like cystic fibrosis. Some patients are carriers for certain genes. So one of the things that we always do with the sperm provider and the egg provider is that we do genetic analysis on them prior, meaning that we look at recessive mutations that they may carry, and if the egg provider and the sperm provider carry the same recessive mutation, then there's a chance that the baby could get the disease. So then we do something called PGD, where we assess the presence of that particular genetic disease in the embryo. Now, we can test for about 250 diseases, and typically we use programs or companies such as Counsel or Recombine to look at these recessive mutations. This is sort of the embryo, this is the embryo development photos. You can see that day 1, you have fertilization right here. You have 2 pronuclei, one from the male, one from the female. That's how we can tell if an embryo has fertilized. So in our program, what we do is we give patients a call or an email letting them know how many eggs have fertilized, and then subsequently the embryos start developing into 2 cells, into 4. On day 3, we should have about an 8-cell embryo, and after 5 days, we get to a stage called the blastocyst stage, which is 128 cells. Now, this is a grade 1, grade quality embryo. If we transfer a single embryo that looks like this into the uterus, we should expect about a 70 to 80% pregnancy rate and roughly around a 70% live birth rate. What are some of the things that make this embryo good quality? Well, there are 2 main areas to the embryo. One is the inner cell mass. The other area is the trophectoderm. This is the group of cells, compact cells, called the inner cell mass. This is the group of cells that actually gives rise to the fetus. So what we look for is we want to see a good compact group of cells here. We measure the diameter of the inner cell mass because smaller inner cell masses are associated with lower pregnancy rates. We also look at something called the trophectoderm, which are the remainder of the cells right here, and those cells give rise to the placenta and the gestational sac. Again, the quality of the trophectoderm, the diameter of the cells of the trophectoderm also have a effect on the ability of the embryo to implant and develop into a live birth, a healthy delivery. So we have objective criteria that we've published on, and with these objective criteria, we can really pick out the best quality blastocysts to transfer. And that's why we're trying to use this in order to limit the number of embryos that we transfer. In count from January to May of this year, if you look at all of our ongoing pregnancies, about 80% of them were single embryo transfers. So when do we transfer embryos? In the old days, it was day 3. Now it's day 5 at the blastocyst stage. We used to do something called day 3 biopsy. So if we're going to assess the embryo for chromosomal abnormalities, we used to take one cell from the embryo on day 3 at the 8-cell stage. But this has become an older technique. The reason is because when you take 1 out of 8 cells from the embryo, there's a potential greater chance of harm. So now what we do is we actually let the embryos grow to the blastocyst stage, to this stage, before we do the biopsy. So this is the biopsy itself. What we have to do is we have to create a little slit in the embryo, and you could see the cells kind of protruding out from the blastocyst, and then the pipette comes in. And then with a laser, what we do is we cut those cells. You can see where the laser is coming in, that we cut those cells, and those cells are taken out. Now, when we do the PGS, when we do the biopsy, we're taking about 4 to 5 cells from the embryo. But there's always a chance of harm. That's why we want to be very selective in the group of patients where we do PGS. Because an embryo that has 128 cells, even if you take 4 to 5 cells out, a lot of these cells have cell-to-cell communication. So you can actually affect or interfere with these cell-to-cell communications once you do the biopsy, and that embryo could stop developing after a certain point. If you look at the literature and the research, anywhere between 5 to 20% of embryos can stop developing when we do PGS. Now, can we recognize that in the lab? Yes, sometimes we can, but sometimes embryos that are that have PGS are transferred back to the uterus, and a patient could have a miscarriage, or the embryo can stop developing, or we may not get implantation at all. So it's not something that's going to develop into an abnormal pregnancy, but PGS has its risks. Now, the research that we're doing right now is we're taking the fluid where the blastocyst grows, and we're trying to analyze that fluid for cells to see if we can do a non-invasive procedure for PGS. So we don't have to create a hole in the embryo and pull cells out. We can actually take cells that the embryo sheds from the media and analyze those for chromosomal abnormalities. So there's a technique called Array-CGH that we used to use before, where we look at complementary chromosomes and we try to match complementary chromosomes in order to assess the embryos. This is sort of a normal result. It's a normal complement of chromosomes. These are abnormal results. You can see on the edges right here, you have either a gain of chromosomes or a deletion of chromosomes. And now what we do is, and this is a complex abnormal embryo because you have multiple abnormalities, either missing chromosomes here or additional chromosomes here. For example, if you look at a syndrome called Down syndrome, trisomy 21, that's associated with an extra copy of chromosome 21. So now what we do is something called next generation sequencing. Instead of looking at complementary areas of chromosomes, we actually sequence the DNA, and we can get a little bit more accurate picture of the chromosomes, including things like triploidy. We were not able to assess triploidy, which are essentially 3 copies of a chromosomes before, but now we can with NGS, which is the next generation sequencing that we are now using. Who can benefit from genetic screening? As I said, typically what we do is we look at the age of the egg provider, and in the studies that we've done in patients who are 35 and older, there seems to be a benefit for PGS or chromosome screening. In patients who are less than 35, for egg donors, in the research that we've done, we have not seen any benefit in terms of live birth rates. In fact, what we did was we did a study looking at a couple of 100 cases of egg donor cycles with and without PGS, and the group of patients who had PGS actually had lower live birth rates. And that's probably because the rate of chromosomal abnormalities with egg donors is much lower, and our system of being able to determine the quality of the embryo under the microscope is really good, so that there's a potential chance that some of those embryos that are good quality embryos, chromosomally normal, we lose based on the PGS analysis. There are also limitations to PGS. When we do chromosome screening and we get a result, there's some limitations, meaning that sometimes the cells that we analyze don't actually represent the fetus. Sometimes you can have an embryo where we're taking cells from the edge of the embryo, and those particular cells may come back abnormal, but the actual cells that give rise to the fetus, which are the inner cell mass, those may be normal chromosome cells. So that's called something called mosaicism that we have to always keep in mind. The other thing is that there are also no tests available for some genetic diseases. Patients often ask, well, I did a transfer of an embryo that was PGS tested and we still had a miscarriage. So either it was because of the fact that the PGS may have caused some harm to the embryo and caused a miscarriage to occur, or the PGS, as you know, has limitations and we can't know as of yet all the genetic diseases or even all the chromosomal abnormalities that are associated with an embryo just with a PGS test. We also do not transfer embryos fresh when we do PGS, and the reason for that is because it takes about a day to get the PGS test results. So we don't want the embryo to linger in the culture media for 24 hours before we have a chance to transfer the embryo back in. Embryos have a window of implantation, meaning that once they get to a certain point of development, they have to start attaching to the wall of the uterus, just like the uterus has a window of receptivity during which it can accept the embryo. And our job as physicians and clinicians is to match the two, match the endometrial receptivity to the embryo. So we did a study in Nevada where we looked at doing fresh embryo transfers with PGS and frozen, and our frozen rates were much higher. So now with PGS, all of our cycles are frozen cycles. As I said, we didn't see any improvement in success rates in young patients, patients less than 35, in patients who had good quality sperm, and of course typically in typical egg donation cycles. So the goal really of IVF and surrogacy is to maximize success rates, to select the best quality embryo, and try our best to really do single embryo transfer to minimize the risks of twin pregnancy, and really to achieve high implantation rates. In order to achieve high implantation rates, if we do PGS, we want to make sure that we have a good cryopreservation, good freezing program, because with fresh embryo transfer, the rates of pregnancy is much lower. This is our ongoing pregnancy rate so far this year. This is 145 transfers from January to May. And this is the reason why we don't need to transfer more than one embryo in terms of improving pregnancy rates, because we've had an 81 rate of pregnancy this year with 145 transfers with single embryo transfers. And as you can see, there hasn't been a significant improvement in ongoing pregnancy rates when we transfer a second embryo. This is 81, this is 93%, but about 70% of our patients who had a double embryo transfer ended up having a twin pregnancy. And what we want to do is we want to really limit the number of twin pregnancies because of the risks to the surrogate and the risks to the babies. And you may have heard just recently about the embryo or the baby with 3 different origins of chromosomes, and that has to do with some mitochondrial diseases, which are very rare diseases, but though some genetic diseases reside in the mitochondria and not in the nucleus. So some of the things that researchers are doing now is to replace the mitochondria or take the nucleus and place it in a donor egg that has a different mitochondria. So you basically are going to have 3 origins. You have the DNA coming from the mitochondria from an egg donor, the nucleus coming from the egg provider, and of course the sperm from the sperm provider. So that's some sort of food for thought for future genetic analyses. Thank you. Yes. No, I think that one of the ways in which you can gauge a fertility center is really looking at their egg donor program, because sometimes what happens is that you have a disparate population when you're dealing with clinics in terms of the different diagnoses. In other words, a clinic may have a higher number of patients, non-egg donor patients, that may have tubal disease. So those patients tend to have higher pregnancy rates. Another clinic may have a lot of patients that have decreased ovarian reserve, which is an egg quality problem. But if you look at the egg donor rates for a clinic, that tends to be a much more homogeneous population. So that's a good way of gauging a particular clinic's quality or pregnancy rates. But clinics typically who have good egg donor and surrogacy rates should also have good rates in patients in non-donor, non-surrogacy cases as well. Yes? No, in both cases, what we're trying to do is we're trying to maximize endometrial receptivity before transfer, and so what we're doing is we're having the surrogate or the intended mother take medications to prepare her uterus, and then at a specific point in time, what we do is we thaw the embryo, we transfer the embryo back in. So the embryo has to be transferred back in within a certain period of time after thaw, and then the uterus only has probably about a 48-hour period of receptivity. If we don't get the embryo back in, then what happens is there's a lesser chance that the embryo is going to implant. For example, that's one of the problems with fresh cycles. So if you try to do fresh cycles where you have let's say an egg donor, an egg provider, and the surrogate, you're preparing the uterus of the surrogate. Now let's say those embryos develop a little bit later. Instead of developing the blasts by day 5, they develop to day 6. What happens is you're missing that window of time when the uterus is prepared, and your rate of pregnancy is going to be lower. So that's why when we compare fresh versus frozen pregnancy rates, oftentimes the frozen are are just as good or sometimes better than fresh rates, because you have more control over uterine receptivity when you do frozen embryo transfer cycles. It's not at zero risk, but there are a couple of companies out there that have expertise in transport of gametes and embryos from one center to another. So we've done that a great deal from different parts of the world within the United States. So typically that's done. Many centers participate in that, where embryos are created in one center and they're transported somewhere else for transfer. Yes. Exactly right. That was with egg donors and surrogates. Depends on age. So if you look at, you look at intended mothers that are using their own eggs, less than 35 versus 35 to 37, 38 to 40. So that's you can find that out from our website based on the age of the intended mother or the age of the egg provider. Yeah. Thanks so much, you guys.
Okay, so some really good questions there. And this is an area where some of this pre-testing can be really important. We had a case in Australia recently where a surrogate was caring for a couple I knew very well, and at 30 weeks pregnancy, the kid was diagnosed with an uncurable disease, and the child died sort of 3 days after birth. It was a very traumatic experience for the surrogate and for the intending parents, and that kind of thing could have been picked up if they'd done the right testing with the embryos early on.